We're now going to hear from Professor or Associate Professor Anthony Gramolini from the University of Toronto. Dr. Gramolini has published widely and extensively on characterizing the cellular mechanisms involved in the regulation of calcium cycling, and more recently characterizing the proteome in the heart. He's a Canada Research Chair in Cardiovascular Proteomics and Molecular Therapeutics, and we're going to welcome him to the podium today to talk to us about whether the genome is in fact enough and whether we're going to have to rely more on proteomics.
Thank you, Mansour. Thank you to the Ted Rogers Committee for this invitation. Like Phyllis, the topic was chosen, and I think I have an opportunity here with some of the earlier discussions not to end up within fighting words of comparing genome, transcriptome. Clearly our interests are proteome. But I think I can weave and bob through some of these earlier discussions that we've just had on genetic sequencing, how things change at the molecular level, and then ultimately to where we're interested in is looking at large-scale proteome changes. And so I think I will weave and bob. We've done a lot of discovery work. We, like many basic scientists, use multiple models. We use cells in culture. We're moving now with Ian Scott and other people to zebrafish models. We've done some stem cell work. There's all benefit and limits to anything we do. We've been doing a lot of work in looking at disease progression in dilated cardiomyopathy. We've done a little bit of hypertrophic cardiomyopathy as well. I'm going to talk about some of these projects. We do large-scale sequencing at the proteome level of progression in heart failure. I'm going to talk specifically about phospholambin. And again, coming through this, I actually was getting nervous through Hugh's talk to make sure that phospholambin stayed within the group of genetically relevant genes. And every slide, I continued to breathe a little easier. And then at the end, as we've gone through the models, actually the mouse models, how in the last few years we've been moving to some of these additional systems. And I'm going to go through those as we go through. But to keep it clear, basic science lab, my group is really grounded in excitation contraction coupling. All of you should have had a couple iterations of this slide in your training. I don't have a pointer, but we can go through. Basically, I am interested in calcium cycling at the level of the sarcoplasmic reticulum. This is our foundation. Anything that changes it, regulates it, tweaks at it, happens during disease, any perturbations, that's of interest to me. And we've now been looking at, really, if we can see changes of calcium release through the ranitidine receptor. You've seen that sort of speckle through the talks. But really looking at the calcium uptake, the SR, sarcoplasmic reticulum, endoplasmic reticulum, calcium ATPase, pumping everything back at the end of a beat. Now, in fact, I've been mostly interested for about 12 years in this regulatory protein, phospholambin, that binds and inhibits the ATPase. We've been doing cell biology, I think, since my second postdoc on how and what regulates phospholambin and ATPase interaction. It was actually a lot of good biochemistry and cell biology. It's a simple 52 amino acid molecule. You can mutate it, study it, stick it anywhere. Look, it's a single exon coding, so before all this GWAS, you could just simply sequence one coding exon, and it worked pretty well. And in collaboration, this was, I think Hugh was saying, in the 90s, that kind of sequencing, together with Litsa Cranius and the Sidebins, we actually had gone through and have now found and been studying 3 mutations in phospholambin that all lead, or actually originated in large families with dilated cardiomyopathy that then we could trace back. It was penetrant all throughout these genetic screens, and identified either a stop codon, 1-point mutation, an amino acid deletion, and then again, we've been studying that really for 10 years at the biochemistry level. The one interesting thing that while we were doing what I consider the conventional biochemistry is I was in the new, now it's the Donnelly Center, but before it was the Banting and Best department, that had all these people coming in, what I think the Rogers is trying to emulate, bringing together a whole bunch of different people with all expertise in different approaches. And in that group, There were fish people, there were proteome people, a little bit of actually large-scale molecular analysis. And we started to actually investigate what's happening in R9C heart failure by mammalian proteomics. And this is now, if you can see, about 10 years ago, and that was new. And so it's true, the mouse isn't the human. But sometimes it's okay. And in fact, the human has early progressive lethal cardiomyopathy, lots of fibrotic infiltration all throughout the ventricle. The Seidmans actually made this mouse and recapitulated nearly everything that was seen in this patient cohort. Early, lethal, lots of pathological staining, decreased cardiac function by echo, and it progressed. So I'm not sure how many people can see, but we have early mortality, about 4 to 5 months of age. Mice live about 2-3 years. And then importantly, the cardiac function decreases about midlife during this progression. So we could study it. And in fact, that's what we did. We took this mouse at this time, basically crudely took out the left ventricular, right ventricular chambers, ground it all up, and spent about 8 months analyzing it by mass spectrometry. Without going into all of the detail, we did grind it up, ran everything, spent, like I said, a lot of time. At the end, we identified and quantified about 6,000 proteins. We could do all the informatics, analyze it to try to find what disease progression actually these big changes are. We found lots of proteins, just over 600, that were changing, maybe not so interesting, but they were changing. We could correlate with microarray. This is the one time we actually had the low-hanging fruit, things that we could really prioritize by proteome, actually does filter through, at least at a transcriptome level, and we can study that. We spent a lot of time, this was new. Anything we found, we're actually, it's probably a misnomer. Proteomics is not really what we do. We really do peptidomics. So we grind everything up, and now we're looking for peptides and then trying to reintegrate everything. And so at this time, we had to prove, in fact, that these proteins changed. And so we went through about 80 different antibodies. It was good. At that time, Canada was reasonably flush. I was flush. We actually could afford to get that many antibodies and do all these experiments. But that actually set the stage that anything we find by proteome actually was being validated by old school Western blots. And I'm not going to go into it, but so this was this intro. We have repeated these experiments. Yes, the PLN is an isolated genetic model. We've done it with probably worse. Jeff Malkenton's activated calcineurin hypertrophic heart. It's basically like a pot roast in the chest. It's so enormous, but it's massively hypertrophic. We've also done the same thing with the progression in myocardial infarction, what happens in probably a more common route to heart failure, as well as an aortic banding pressure overload. These aren't so simple, but we've been trying over the last bit to integrate these for common pathways, irrespective of cause. We've actually taken a bit of this data and tried to look for, can we take things that we identify on large scale and identify them as markers during disease progression? Surprisingly, we've had some success for moving that forward. That actually is at least identification by mouse studies of heart disease that actually translate a little into patients. But I agree with the earlier talks. Our discovery for those years had been, I think, bottom up. We've been looking really at the mouse. The mouse is not the human. The mouse beats, on average, 700-800 beats a minute. I don't know any amount of stimulation in a human that can get anywhere close to that. So there are fundamental biochemical differences. So we were very aware of that for at least, well, we've always been aware of that, to be fair. But how do we address it? So we've really been looking at these proteome changes in human. Phyllis said it's a fundamental issue. How do we get human tissue? Especially, how can we get healthy adult human tissue? It's a challenge. Phyllis indicated we probably at this time can get, I think she referred to it as flex. When we did these experiments, we would need the equivalent of a thumb. So it was a challenge. We came at this with a couple different approaches. One we actually just wanted to set the stage. What is an atria? What is a ventricle of a human tissue? We actually were able, with Bob Hamilton and sick kids, to get some human, healthy fetal tissue. I'm going to want to walk through that. And then even as we've gone past that, after we've done chamber, how do we get to the real, the unique proteins? So again, really simply, we can take tissue. I'm happy to talk about all the methods of how we actually analyze it, look at it, separate it informatically, group it all back together. But in effect, we can take these human fetal tissues, separate them biochemically, analyze them in great detail by peptides, reassign everything back at an informatic computational level, and then begin to see what these proteins are in human tissues. Now, if we look on your left, that we actually can go through human tissues and find what actually were all the proteins at this stage that we could identify in a human atria. We can do the same thing, human ventricle. Almost this year that we were publishing this work, there were 2 different studies. We were beaten. 2 big studies came out and looked at a whole bunch of tissues in back-to-back papers in Nature that actually looked at various different aspects of what's a whole fetal heart, what's an adult post-mortem, about 8-hour autopsy sample. And then we could actually begin to look at, between all these studies, begin to assign what makes a heart. It's not just troponin T, it's not just myosin heavy chain, myosin light chain, maybe some of the desmin related molecules, but we've actually built this entire study that we've actually gone, and I think we have a good handle on what at least fetal tissue is an atria and ventricle. So if we really get into some of the real detail, everybody likes to see individual genes. Mathematically, I'm happy to talk about any of these. However, you really clearly see that we have proteins that are only ever ventricular, only ever found in the atria, some that are differential. And again, maybe you can't see further back. I've put a little asterisk. Within here are all those known ones that are candidate genes for disease, candidate markers, ANF, troponin T, myosin heavy chains. But we found, I think, hundreds of other highly abundant proteins, which now got us at this time really excited that this actually is working. We can identify chamber specificity. At least these now are fetal tissues. Can we extrapolate that moving forward? At the same time, I won't really get into fighting words. We actually are, I think Mansour said, risk averse. We actually have parallel microarray. It's not perfect, but we can actually use that as a strength. Where there's convergence, we did it. We have parallel microarray, go through it all. We can analyze them independently. We can analyze by convergence. Here we actually have the independent microarray. It's great. This actually very stringently, 9,000 genes, kind of fits in this, the juggle, what we think would be expressed in any individual tissue. We could go and identify uniqueness. That's fine. But we're interested in now some of the highest abundant convergent at the protein and transcript level that are actually unique. So we have in here, again, I think you can see some of these that are probably, if you just see the little asterisks beside them, some of the ones, some of the proteins that are known to, at least by developmental studies, general biology studies, that are uniquely expressed or differentially expressed in a chamber. Now maybe if we look on more the ventricular side, a number of these are the candidate genes that we've been seeing most of the morning. Myosin heavy chains, probably the troponin T, troponin I is in here as well. But we have other things, other proteins that now clearly segregate at a transcript level, protein level, that actually, in my opinion, should be candidates for at least growing into these not to go back, as Hugh says, study more genes, but they might be of relevance to actually begin to consider as we go forward, at least for some of the clear phenotype disease. So we've now put this together. This is of interest. This, I think, was a good exercise for us to do. It's older data, but actually, where does it go from here? Most people, actually, we've seen, at least Phyllis is talking about IPS. We'll see other people at SickKids talking about IPS lines. Well, I think we've set at least human fetal tissue is a good benchmark to aim for. These tissues now for chamber specificity and differentiation. We are a lower organism basic science lab. We will study these molecules as they contribute to chamber development, ventricular disease, ventricular function in general. And so this is some of this utility of just how do you separate a heart is not just a heart, but we have some things going uniquely. So no sooner had we been doing these experiments, and as we continue to talk, the heart, the ventricular wall, is just not all myocyte. I mean, I'd started before, we all think about that. We're all trained to think about that sarcomere at the EC coupling. But in fact, there are other major constituents in the heart that aren't myocyte. And we've been thinking, at least as we go forward, that it's not myocyte. We have multiple chambers. And here, what I'm actually really coming in to say is that at this point, we were also addressing, in this whole talk, the genome, transcriptome, proteome. We have a number of issues converge for us that, I guess in the last 5 years, there's been now about 3 studies. It's extremely difficult with people to do high-end proteomics who also want to do transcriptomics. But the general consensus is transcriptome has utility, but the correlation to just even simple protein changes is actually, being fair, modest. So, and it's modest for a lot of reasons. I think if you can see, we have standard, now if we have RNA-seq data or general transcriptome data, you can assess peptides wishfully. You could identify small DNA sequences and look at what some of the genes are. I'm already wanting to get already to the protein. But they're useful. But past the transcript, that protein gets translated. It has a localization. It's probably regulated by phosphorylation, tagging, any other post-translational modification, and it has a half-life. That doesn't get taken into account in any transcriptome data. And in fact, the few studies that actually have tried to correlate any condition where there's a change in a simple organism at a transcriptome and a proteome has done it in extremely simple systems, yeast, and the correlations, if you can see or not, Pearson, Spearman, however you do the math, these are the most abundant studies as they've gone in and done it, I think, carefully, actually only have about a 0.6 correlation. So things that change, it may work for your gene as a one-off. It may be fantastic. But at a global level, it begins to unravel. And so for us, on top of everything that we know with our chamber specificity, how we're moving, we can't do this by transcriptome. Actually, I started by saying I'm the most interested in the sarcoplasmic reticulum, which is populated by membrane proteins and regulated at the plasma membrane by membrane proteins. We do know that the transcriptome has probably the most dysynchrony to the proteome for membrane proteins. So that we know. So we couldn't do, it's not rewriting history. We just couldn't have done moving forward by a transcriptome. So we actually spent, now this is about our last 5-year program, is to take, how do we go now and look at the cell surface and the membrane proteins of heart tissues for proteome work to get to really where we want to now overlay to disease, look at heart failure, look at hypertrophy. And so we came up with some simple old chemistry with just charged small glass aluminum charged beads, put them on any cell, isolate off biochemical membranes. And we did this with neonatal mouse cultures, but those same ventricular tissues that we would get the human fetal tissue from the ventricle. And then now we can go and get the cell surface and membranes out of mouse and human tissues. We can do biochemistry, I won't go into it, but we actually biochemically really know that we are isolating membranes and everything else. We can do lots of mass, I'm going to open this all the way up. We can open up lots of mass spectrometry, lots of number crunching, taking a mouse ortholog to a human ortholog, that they actually have to be the same family member, crunch that all together, come do more statistics. But I think if we look at all these colored bars, that at the end, we've actually, through this study, identified just over 500 proteins that we are interested as membrane proteins that show conservation between mouse and human, that we've added in red. You probably can't read it. We can do an informatic analysis of these 500. We selected things that were not studied. So we have about only 20% of our proteins that we're looking at out of these 500 that actually have a literature assignment to cardiac or muscle or anything that you could actually say is a heart. So in fact, what we've now identified is about 350 or 400 proteins that actually have conservation between species that are membrane proteins that haven't been studied. So we got excited. We've just published our first study off of this one candidate, our number one. So we have 500. We can do lots of informatics, basically churn data, and at the end have a rank ordered list. And our rank order basically came in, and our number one candidate was this transmembrane protein, TMEM65, that's clearly ventricular enriched, almost exclusively heart enriched, as well as other proteins as we go through. We're going to hear one of the grad students tomorrow talk about the number two candidate as how he's worked up that candidate. But we focus here on number one candidate that we identified conservation, looks good. We took advantage of now actually checking in human tissue, human heart tissue, for lots of these proteins. Are they there? Are they in the cell that we think they should be? So TM65 we know is a gap junction protein. We know exactly what it's, well, we think we know exactly what it's doing. But we have lots of other proteins. Discoidin, PopDoc-DC2 was a Popeye domain-containing protein that's just had about 3 papers in JCI and Circ, linking it to arrhythmias in the last year and a half, as are other proteins. So we feel very good that this is actually a discovery tool. It's basic science, but it was a discovery tool. One of the things, though, that we were immediately aware of, linking that it had to be mouse and human, actually made our job simpler. Clearly, you're not going to see the text of this, but I think you can see that the convergence at the very top of the sequence of human, all the way down to C. elegans, is extremely high. So again, just by adding that we had to identify it in a mouse and a human tissue allowed us to look for, I think, again, low-hanging fruit, that these are novel proteins that have been conserved in evolution, allowing us to initiate an entire set of studies with additional model people within the Greater Ted Rogers Center, and we're going to hear about that with Ian Scott tomorrow, that we actually are now able to make models of some of these proteins looking at development based off of these high degrees of conservation. And as, again, many of you might not appreciate, membrane biochemistry has its own unique challenges. It's not a straightforward set of biochemistry because, again, these are all within lipid membranes. And in fact, we've been doing biochemistry, structure function studies. And again, I think the model systems have been useful. So as we go through any discovery, these are new proteins. It's taken us actually about 4 years to work through TM65 itself, that where it's expressed in, this is an isolated adult mouse myocyte. In the middle is a mouse cardiomyocyte section through a ventricular wall. We've done, again, with sick kids, immunogold labeling to show exactly where this particular protein is at an intercalated disc. We've been moving forward a little. This, like we've seen the earlier slides, TM43, at least an ARVC, ARVD candidate gene. We actually do believe, we looked, Hugh had indicated that the GWAS data and the data out there is not very robust. We actually took all of our proteins that we actually really think that these will be involved in arrhythmias, the things that we see at the intercalated disc, things at the cell surface will likely have other congenital defects as well. But we can't make that linkage yet. So I think one of the things looking forward for us is to take all this data and actually begin to tie in with the genomic sequencing efforts at SickKids. So we think we've been okay. But I think every step that we've gone, we've appreciated, well, did we really actually do what we should have done? Most of all of our, again, I made the analogy that we would take out a heart. We all do it. Any tissue, you take it up, grind it, you do transcriptome. You take it up, grind it, and you do a proteome. The heart is not actually a homogeneous tissue. And in fact, we now appreciate that. We've done the exact same, we're a bit repetitive, we've taken the exact same proteome approach, and now look, though, at human cells. human cardiac-derived endothelium, human cardiac-derived, coronary artery-derived smooth muscle cells, human ventricular-derived fibroblasts, and basically did this same big analysis at the proteome level and looked at all of these proteins. All the informatics, we can churn data. I think we've gotten very good at churning data. But at the very end now, we have, I think, a very clear picture of at least what the source cell in a human heart for the fibroblast proteome, the cardiomyocyte proteome, the endothelial cells, and the smooth muscle cells, now I think allowing us to go moving forward. So coming back to this human cardiac tissues, if we look on the left, there are very clear proteins that we know in a cardiomyocyte. I don't have the fibroblasts. We have cardiomyocytes, smooth muscle cells, endothelium, fibroblasts. Some known. We've actually gone and started to look at all these proteins that are very unknown, unlinked, that actually are clearly either lining the entire myocyte cell surface. You can see the top two on the upper part, new proteins that are linking into the gap junction, or just even new proteins that are uniquely expressed in the vascular beds. So where have we really gone now? Trying to come all the way back. I think we have a pretty good picture of the human heart, what makes that human heart, how we break it down and reconstitute it. Surprisingly, I wished we could be myocyte. Actually, most of the changes we see and everybody else actually carefully looks through now these big changes in whether it's dilated, progressive, hypertrophy, are fibroblasts. Actually, our big data showed us that, in fact, the predominant protein change of all of our proteome were fibroblasts. So we went ahead and actually did an infarct model in a mouse, went and took then, you can see vimentin is a marker of fibroblasts in green. Actually in that, this is a very big infarct, but you actually see fibroblasts in that infarct. F-actin, a marker of the contracting myocytes in red, is almost gone. So in that infarct, we have lots of fibroblasts, no myocytes, and all of our fibroblast-enriched proteins that we've identified through all this big study actually are lighting up this infarct zone. So I've said within our greater group, I go, actually, naively, it always looked at that red that you see in the infarct and actually viewed that as a dead scar. But in fact, it's not really a dead scar. It's active live fibroblasts where we've just lost the contracting myocyte. So we've had discussions within at least TBEP, the Toronto division of the Ted Rogers Center, is can we recapitulate around understanding the degeneration and regeneration at this scar that probably is all fibrotic. So where are we at the very end? If I put all at least our proteome data, we've done a lot of trolling. But that trolling now at the very end has led to, we know in the human heart, all the proteins that we can identify, about 6,000, which I think is close to that transcriptome at least. We know which chamber they're in, and we know which cell type they're in, and for us, whether or not they're a membrane protein or not. So we've ranked them, and I think we feel okay, but we at least have that, I think, in our tool shed moving forward. And that's where leading ahead with the combined, I'm excited about the Ted Rogers Center. As we are excited and moving forward, we actually now have, I think, a good framework for this heart, at least in early either development or early stages of health. As we go ahead, we need to understand what's happening in congenital disease. We're going to look at some of these models as we go through how many of these proteins actually are causing early development of either individual chambers or individual cell types. And so on the bottom, I mean, we're really looking forward. It's a data set, but we've really been spending probably the most time on identifying new proteins in cardiac function, how we can go after them, what they actually are doing, with the greater understanding that we have to apply this back to, maybe not just back to the phospholamid mutation, but at least what's happening in infarction, what's happening in pressure overload, what's happening in earlier congenital cardiac diseases. So with that, I think that's kind of where I end on the proteome work. And thank you very much. We've had a lot of people working together, some stem cell, some conventional biochemists. And thank you.